scratching and kicking and biting you. So two people wrestle it, and you hold it in place, and then somebody comes along. Usually, we used to get a little shot of anesthetic to calm it down, and then we were doing things like taking blood samples and milk samples, and we were checking the pouch for joeys and measuring the joeys and stuff like that, looking at the teeth, and then we would wake them up and let them go. And then we go off and find somebody else to hatch that in the evening. And we do that for about 4 or 5 hours in the middle of the night. And it was a lot of fun. we had a driver, we had a shooter, we had someone who sat on the top of the four-wheel drive with a big light, and then we had two people on the back, which were runners. And I was never running because I was too sluggish. So there were two fast guys who jumped off the back and ran after the kangaroos after somebody had a shot at them and somebody's jackrabbit and stuff. And so it was definitely insane. Yeah.
Did you ever encounter any of those ones you see that are like real muscular and want to fight?
We actually avoided it because we were looking for the girls.
Okay.
So we were looking for ones with joeys and we were interested in that. So yeah, we kept away from those. I've seen them sitting back there taller than me. I couldn't look them in the eye. The big red kangaroo. So I would not mess with one of those. But the females bite and scratch and growl at you like a lion. It's really disconcerting because they look so cute. But when you've got them in a headlock, right, they're looking at you with like the stink eye and they're growling at you, and you got someone else hanging off the bed as well. I should try that. But when they had joeys, when we let them go afterwards, we had to use masking tape and we had to tape up their pouches because when a female kangaroo is stressed, when she's chased by a predator, one of the first things she does is reach in, grab a joey and throw it over her shoulder to the predator because she can have another baby within weeks. She's got a young at foot running with her, one in the pouch, and an embryo that sits waiting for one of the teeth to become free and immediately give birth and attaches again. So when times are good, they can have 3 constantly all the time. So one joey is better than 5. So we would have to put masking tape on the because so when she woke up and started panting, she started scratching at her pouch, and so she couldn't get in there. And then when we drove away and it goes all dark and she calms down, then she would just like pick off the tape and then everything's fine. As far as we know, anyway, they didn't. She didn't need that child anyway. You know, so yeah, that was interesting time. We did that for a couple of years. There was a young American student doing a PhD there, and she was in charge of that project, and we were just helping her with that. That was a lot of fun. Was that a good sound check?
Yeah,
is this good? Is it best with me using this? Or?
Yes.
Okay. All right. I'll try and remember to do this, right? Yeah, I was a pretty good shot, so I used to be a shooter. And was that going to work? Oh, good. It was going to work. Yes. Okay. So, yes, I was a shooter for a while, and I used to clip ears occasionally. Never hurt them badly, but used to clip ears. And they had big ears. They ears like donkeys. They're huge. Right? They're like 8 inches tall, and I sometimes clipped blood vessels, and we had to patch them up afterwards. Because the area you're aiming for is only about a 3-inch square area just above the head. And, but I never terminated any. Always being an error on the cautious side and aim up a little bit. It was fun. Am I on time now? Am I ready to go? Okay. Looks like I'm not going to have enough hands, so I had a little. I don't normally speak with notes, but I had a few facts and figures that I was going to give you that I probably can remember. If I can't remember, I'll pick it up. But normally I don't do this, and I don't normally talk with a microphone in my hand as well. So if it drops away and somebody can't hear me, yell at me. Okay, to do this. Firstly, I want to thank everybody, and especially Mary and Lycos people for inviting me to CrowdCon again. It's one of the highlights of my academic year. to come here. It's great that it's in the summer, so I can actually devote the time. When I'm teaching, it would be hard sometimes for me to get away. And I really enjoy coming here and chatting with you all. And now after a couple of years, I know a lot of you, and it's so much fun to come and hang with a community of people who are interested in crabs, probably more so than I am to some extent. I have a really broad interest in crustacean crabs, and it's nice to come to a group that's very focused in and what they're interested in. But my talk today, I'm going to actually broaden your interest out a little bit more and talk a little bit more about the relationships of land hermit crabs, both within the species of them, but also who they're most closely related to, and sort of put them in context of crustaceans for you. And hopefully a lot of it's new for you, and if it isn't, then I'm sorry, you'll just have to bear through it. Don't point at the screen, point at the machine. Okay. So basically, it's a family relations talk I decided to give today. Kind of set the scene to put land hermit crabs into their family. And I don't mean family in the taxonomic sense. I'm going to talk a little bit about that in a few slides, but just like their relatives, so to speak. And there are conventional families and there are unconventional families. And here's a bunch of hermit crabs you'll recognize. Oh, now I need another hand. Oh, that's a work. That'll work on this. Good. I put my other green pointer. But did I do that? Because sometimes using the pointer on this machine. Did I do that again? Yeah. Okay. I can't use the pointer on this machine. You're going to be in charge of slide changing. I'm going to use my green pointer. Can we get back to full screen? Yeah. I use this all the time in teaching, but I have a Dell computer and not a Mac, and sometimes there's issues with that. And even if though you're using the pointer, it's changing the slides on you, so it's kind of weird. Okay, let's get back to these images here. Here's a bunch of hermit crabs, and these are all hermit crabs. Of course, you'll recognize your beastie here, right? In light of the land hermit crab. And then you've got classic marine hermit crabs living in shells like these guys. And then you've got a bunch of hermit crabs, and yes, these are all hermit crabs. This is the deep sea one that doesn't have a shell at all, or this one actually, we have to see that well, actually carries an anemone on its butt, no shell. This is a hermit crab that doesn't live in a mobile shell. It's permanently buried inside a living coral on a coral reef. So it's stuck in that hole for its entire life. This is a symmetrical hermit crab, which there are some. We'll talk about those later. And this guy here is actually a hermit crab, although we would recognize it as such. We have a common name for them as being a stone crab, and I'm going to talk more about them later. And that's not the Florida stone crab. This is where common names don't mean anything. Not the Florida stone crab, but the stone crab because that's what their name means when we get to that. Can we get that next slide? So this is more like the family. It's not the Brady Bunch for hermit crabs. Right? It's an unconventional family, and there's some very different looking relatives, both close and distant, in this group. And so that's what I want to talk about today. Okay, so what I'm going to do first is I'm going to start off close in, and then take a step out, and then take a step out again, and sort of place your land hermit crabs in the hermit crabs within their larger group, and then put them within the crustaceans so you can get some idea of their broadening relationships. So we'll place the hermit crabs, the true hermit crabs, in relation to some of their hermit crab-like relatives. And next slide. And then we'll put the hermit crabs here and the true crabs in relation to a whole bunch of other crustaceans. It's not important for you to see the details there. This is just what I'm going to do. I'm going to build out a little bit as we go. Okay, next slide. Also want to acknowledge that one of Moa Lundberg's, one of your community photos from past years, she takes some fabulous close-up portraits of her crabs. I'm going to start off somewhere. I'm going to give you a quick minute lecture and reminder about the taxonomic system that we use for naming things, because I'm going to be throwing names at you. The typical Linnaean classification names that we use, right? Like species, genus, family, order, phylum, etc. That Carolus Linnaeus, as he was later heard, his students loved him so much when he died they Latinized his name, which is what we do for all scientific names today. So he started off as being Carol Linnaeus. He was a Swedish botanist and a Swedish physician, and he invented our naming system that we use today in 1758, and we still use it today. And his system starts off with the most inclusive groups at the top, which is the kingdom level, and then it comes down through phylum and family, order, genus, to the most exclusive group, which is the species level. And the species and genus combination, and I like to use the analogy, it's like our family and given name system. So in your family, there are lots of Smiths, all right? Daddy, Mummy Smith, and sibling Smiths, and you all have a given name that goes with it to set you apart from your siblings and your parents. So we can think of that, the genus name, as being like a family name. So we have all of the members of the genus Cenobita, and then we have all the different species, which is almost like putting a given name onto it. We have Cenobita variabilis and Cenobita rugosa, and Pseudorugosus and Pipiatus and Compressus, etc. And that's the most exclusive level. There's the least number of individuals in that box. And the greatest number is at the top here, where we have what are called domains at the top. It used to be kingdoms, kingdom Animalia, the animal kingdom, kingdom Plantae, the plant kingdom, and fungi and bacteria. Now we have domains above that, and that's the most inclusive, where we are in the domain with everything except bacteria, which are in their own domains. And in fact, bacteria have two domains that they're split into. Everything else fits into a third domain. So that's the most inclusive level, and it becomes the most exclusive as you work your way to the bottom. And then this is for the black bear down here, actually. And then on the right here, I put the same sort of thing for our crab. So we have phylum, where it would be chordata. It's arthropoda, the arthropods. Then we have, that's class there. So class Mammalia, where the mammal would be for a bear. It's class Crustacea. And then we have order Carnivora for the bear. And then we have Decapoda for the crab. I'm going to be working from this level down for most of my talk. Family Ursidae for the bear. Family Cenobita for the crab. Genus Ursus, genus Cenobita. And I think this is Ursus americana. Yeah, that is. And then we've got, what did I put there? Skittle or something like that at the bottom there. Okay, so that's the level we've been looking at. And I'm going to be using names from kind of this level down for the crustaceans. So I just wanted to remind you about that. Next slide. All right, this is also hard to see, but I want to also talk a little bit about the distribution of Cenobita species. They are a tropical species, found mostly between the Tropic of Cancer and the Tropic of Capricorn, 30 degrees north and south of the equator, with some of them, they're basically split into two groups. The one group ranges from East Africa through Indian Ocean into Southeast Asia, Northern Australia, down through Micronesia, all the way out to, I think the most easterly place is Pitcairn Island, I think is the most easterly point that they've found it. The most northerly ones are some of the Japanese species from the southern islands of Japan, the Ryukyus and those sort of places. And the most southerly are found on the Australian coastline, down on the western side of Western Australia and on the Great Barrier Reef here. And then you've got a second bunch with a lot less diversity, which is over here in the Americas, where we've got Pipiatus and Compressus over here. I saw some records down to northern Chile. I don't really know if that's true or not, but definitely they found Ecuador, the Ecuadorian crab, right? The Pacific coast and the Atlantic coast, Gulf of Mexico, and the Florida coast here. And then strangely enough, there's one species right here in West Africa, but we're going to talk about that at the very end when I'm going to talk about questions about distribution and how do you get crabs all over the world like that. But let's just say the greatest diversity is here. The greatest number of species is in Southeast Asia, into the Indo-West Pacific, second smaller group over here in the Americas, and they're pretty much between 30 degrees north and south. They're tropical animals. They don't like cold water. Okay. Next slide. Okay, so we just had a lecture about Bogus latro, coconut crab. This is Cenobitis' closest relative. It's the only other genus in the family Cenobitis, and it is essentially a shellless land hermit crab. Okay, it happens to be. It was named by Linnaeus himself in 1767. Somebody must have brought him back a crab specimen from somebody traveling in the Indo-West Pacific. Brought it back to him in Sweden, and it was described and named by him. And it can, that's there. It's also called the coconut or the rubber crab. It's coconut crab because it eats coconuts. It was called a rubber crab by many people who come across it because it will steal anything that's not nailed down. I've seen photos of it dragging pots away from campfires, camp chairs in its claw, carrying empty bottles of rum up trees. So it was called a rubber crab as well. Golf clubs, yes. We must have a wicked swing. They can grow to be 10 pounds in weight and 3 feet in length from the tips of their claws to their tongue. So we're talking about a significant, large individual. You can see some of them here. Look at this. This is a standard trash can that this one's gripping to here. So it is the largest terrestrial arthropod that's alive today. All right. It's not the largest terrestrial arthropod ever. That goes to a 2-meter long fossil millipede called Arthropleura, and 2 meters is 7, nearly 8 feet long. So that is the largest terrestrial arthropod. This is the largest living terrestrial arthropod they found. It's also the largest living terrestrial crustacean. It's not the largest crustacean. Who knows what the largest crustacean is? Aquatic or king crab. It's the Japanese spider crab. Can I see the next slide? The Japanese spider crab. Okay. Macropura tempori. It can be 13 feet across when the legs and the body can be the size of a basketball at their biggest. So this is the largest crustacean in size. There's different ones in weight and stuff like that, but this is the largest one in pure dimensions. The largest aquatic crustaceans. Can we go back one? All right, but this is the largest terrestrial crustacean and heaviest terrestrial crustacean, and the largest living arthropod on the planet today. Okay, but remember, even though it's running around like this, these are 2 little baby burgers in their shells. They still live in shells. When they drop out, their usual life cycle you've all heard about. Planktonic larval stages, go through zoea to a megalopa. Megalopa drops out of the water column onto the bottom, finds a microgastrified shell, and crawls into it, and then spends a short amount of time as a benthic or bottom living shelled hermit crab, and then one day walks out of the sea onto the beach. Drops the shell and never goes back. Okay. Why would Burgess not need a shell or use a shell? No predators. Could not have any predators. That's right. Traditionally, they're mostly on isolated islands in the Indo-West Pacific, where until man turned up with dogs, cats, and rats, okay, to kill and eat them, and when man started to eat them, they didn't have any large terrestrial predators on this island. And so if you don't need a shell for your support, for your protection, then you can actually grow big enough. And that's another thing. If there's no shells restricting your size, then you can grow bigger. You can grow to be 3 feet tall. If you have to fit into a conch shell, right, you can only grow as big as the conch shell. If that's the largest shell available to you, then that's all you're going to have. And so not only is it a protection, you can grow big because nobody's eating you, but it also means you can leave your principal restricting resource behind, which is your shelter shell. And that then frees you from the restriction of size that you need. But then, of course, you leave the shell behind, you leave your protection behind, and then when you are later introduced to predators, those predators have decimated natural populations of Virgus latro throughout their range. They're extirpated, I should say, on many in northern Indian Ocean islands and many of the Indo-West Pacific islands. They no longer occur. They were eaten out by man. They were eaten out by dogs and pigs and rats. And so they're only on certain islands where they've been able to eliminate those, and that they're able to you're able to find them in their native surroundings and wild surroundings. Okay, Karen. Next one again. I must have duped it. Okay, so speaking about hermit crabs that don't have shells, let's go back and look at a bunch of hermit crabs that also don't have shells. These two images were like the one I showed you before. This is a deep water hermit crab, and I mean like really deep water hermit crab. This is called Providii, genus Providii. This guy has a naked little butt on the end and nothing on it, but sometimes they pick up a large anemone and they stick it on their butt and they carry those stinging tentacles around with them, and that's part of their protection. If something comes up to bother them, they just swing their butt around and point the tentacles at it, right, for some sort of protection. Okay. There you go. So this is both Providii. They can be without it, or they can be with something like that. Here's that Pagurata, the stationary, sedentary, tropical hermit crab that lives embedded in usually massive corals. High corals are ones that have globular or don't have fingers on them like this, and they usually find a hole that somebody else has already made, or when they're a larva, they drop into a tiny empty coral cup, and then they sort of take it over, and the coral grows around them, and then they have this permanent little burrow that they live in. And as you would expect, because they can't roam around, they're restricted in what they can eat. And so these two long antennae here, you can't see them in this picture, they're feathered, and they use them to catch plankton. So they're plankton feeders. So they stick there, have the coral, and they wave their little tentacles around, and they grab plankton, and they lick all the plankton off, and they wave it out around, and if danger comes, they just pull back into their little hole there. And they'll often occur in male-female pairs within a few inches of each other on the surface of the coral, so they must at some point, one has to leave their burrow to go to the other one to reproduce. And then here's one here down here called Cancellus, and it's hard to see there. That actually has a stone on its butt. It picks soft stones, things like pumice and soft sedimentary stones. It actually excavates a hole in it and sticks its butt in there and carries that fairly light rock around with it. And so it's using a large stone, a soft, light stone, but a stone instead of a shell. And then here's Pilocheles, two of them in there, and they live inside wood and bamboo that they find at the bottom of the ocean. And they excavate. They don't have to excavate in bamboo, it has a natural cavity, but into the wood they excavate a hole, and they literally carry a small piece of wood around with their butt embedded in the end of it, or a piece of bamboo around with them, again, wandering around. So I send the same question out to you as I did for Virgus latro. Why would these hermit crabs not need shells?
Resource.
Good answer. Many of these, except for this guy here, are all deep water hermit crabs. And once you get below a certain depth, with the increasing pressure, calcium carbonate shells dissolve. There are very few gastropods at 3,000, 5,000, 6,000 meters at the bottom of the ocean because they can't keep pulling the calcium carbonate out of the water and making their shell and keeping their shell intact. It actually dissolves. So some snails are down there. They actually have reinforced their shells with metal like iron and molybdenum and stuff like that. They live in a reinforced shell because the calcium carbonate dissolves. So if you're wandering around at depth as a hermit crab down there, you're not finding shells. There's none down there. If a shell happens to drop down there, it can dissolve in a few years and completely into the water column. So there's no shells. So what do? You find things that are really common down there. You find bits of sunken driftwood. The bottom of the world's ocean is filled with driftwood. Right? All those trees and branches that flow out of rivers into the ocean, many of them sink to the bottom. And there's an entire suite of organisms that live off that sunken wood, including polychaete worms and echinoderms like sea urchins and stuff like that, that live in and on that sunken wood. And these guys take advantage of sunken bamboo and sunken wood and can sell us as well. There's plenty of rocks in the bottom of the ocean, right? If there's no shells, and so they're taking advantage of that. And then Pangurina is a special case where, like many tropical marine organisms that can live in holes in corals, things like Christmas tree worms and other types of polychaete worms and stuff, they're just taking advantage of the fact that they can live in a living organism that can protect them, and they can get all the food that they want from the water column, and so they don't need to be mobile. And they've given up scavenging and moving to take up a plankton-eating sedentary lifestyle. So, shells are a great resource, but there are some places where they're just not available or just not needed. And so multiple times in the hermit crab family, they have abandoned the use of gastropod shells, either by choice or by necessity. That's another slide. Okay, so typical hermit crabs here. If we're going to talk about Cynobita, in the genus Cynobita, who are they most closely related to? And you can't see the details here, but on this phylogenetic tree, which is looking at the relationships of hermit crabs and their relatives based on genetic data, and I'll talk about that in a moment. Here we have the Cynobita hermit crabs, and next to three of their closest relatives here, which are the Diogenid hermit crabs in the family Diogenidae, which are the left-handed hermit crabs. And here are some of those representatives here. We've got Clibonerius, Diogenes itself, the type genus, Dardanus, Taxonomy, Ciliopagurus, and that one is Isocheles. Okay, Isocheles, thank you. So they are all members of the Diogenidae family, and in any trees that you create, no matter what the data you put in, whether it's genes or whether it's morphology, the Cynobita land hermit crabs always come out as the closest relatives to the Diogenes. All of these guys are marine. There's no other terrestrial hermit crabs other than the family Cynobitidae, but their closest relatives are the marine hermit crabs in the Diogenes. Some of you might say, what? Left-handed hermit crabs? There's a left-handed hermit crab? Next slide, please. Hermit crabs can be basically broken down into two principal groups. The ones that have a slightly larger or very larger left claw, or the ones that have a larger right claw. And this is Dardanus here, typical Diogenid. All the Cynobitas have a larger left. Now, the only time it won't be like this is if it's dropped a claw and it's trying to grow it back, right? But it'll always have a larger left claw. So all of the Diogenids I showed you, right? Can we go back one slide, please? You can't really see them in the slide here. All of these guys have a larger left claw, or they say they're sub-equal, and if you measure them, the left is just a little bit bigger than the right. That's why we have a genus called Isocheles. Cheles is the cheler of the cheliped. Iso means they're the same, so it's called Isocheles because its two claws are almost exactly the same size. So they're either sub-equal, or the left is a little bit bigger, or massively bigger. Can we go forward again? Or the Paguriid hermit crabs and another family called the Parapaguridae, which are the deep water Paguriid crabs, all have larger right hands. So you can basically split any of the aquatic hermit crabs into the Diogenids and Cynobitids, the left-handed hermit crabs, and the Paguriids and the Parapaguriids, and the Lithodids, the stone crabs, we'll talk about that in a minute, which are all right-handed hermit crabs. Go to the next slide. And this is how they fall out here. So here's the left-handed one, family Cynobitidae, family Diogenidae. These yellow ones are new families that are recently being suggested by a guy in Europe who studies fossils. One of them, the Calcinidae, is a genus within the Diogenidae, and he's arguing that it should be its own family. And the other one there, which I can't pronounce so long, is suggested from fossil evidence only. There's no living representatives of that group. So anything in yellow is newly suggested families. The The ones in white are the standard families that until this guy had made the suggestions later. And then all the right-handed hermit crabs, the figurids, the Polychaetididee, the Parapigures, the Lithotids, the Haplognaths, and then these three groups here who recently. And then right at the bottom here, the Polychaetididee, you remember I showed you a symmetrical hermit crab, the one that lives in the bamboo and the wood, okay, has a symmetrical butt. So when you pull it out, it looks like a crayfish. Right? Its butt doesn't curl around, it's all calcified to the end, and it looks just like you pull a little crayfish out of it. They're the symmetrical ones. So you have one symmetrical family, there's about 5 to 8 or so right-handed hermit crabs, and then there's between 2 and 4, depending on who you believe. These yellow ones haven't really been kind of vetted by the community yet or accepted. It's really difficult for people who study living crabs to incorporate fossil stuff into their thinking. And of course, anyone can have opinions about names and groupings, and it all depends on what the community is going to accept and what becomes commonly used. So these are all being published in the literature. They're all good names, but whether people are going to use them or not, it's going to take a little time for that to come about. So the left-handed hermit crabs, the right-handed hermit crabs, and the symmetrical hermit crabs. And so your land hermit crabs are left-handed, and they're most closely related to that family, the Diogenes, which is all aquatic. So obviously, Cenobita is probably a derived Diogenid, or had a derived Diogenid ancestor, and is the only one of all the Diogenids that decided it was going to take up living on land. The rest of them have stayed in the marine environment. Okay, next slide. All right, here I want to do 3 tangents to cover a couple of things that I just talked about. One, I want to talk about trees, because we're going to see a few trees, and I'm going to talk about the difference between molecular and morphological trees. Phylogenetic trees, I mean here, which is those trees of relationship. It's a bit like a family tree. We always have them this way. We always make phylogenetic trees this way. I mean, when we draw family trees, we tend to draw them this way. Okay? I'm going to talk about asymmetry in hermit crabs. We just talked about left and right-sided and symmetrical, but I want to talk about the general asymmetry that all hermit crabs have and why. And then lastly, I want to talk about this very special group I showed you, the Lithoted hermit crabs, which don't look like hermit crabs at all. Okay? And so let's investigate these three tangents here. So here's a great big molecular tree that somebody published recently. It's based on 4 genes, and it's got the family Cenobita is right here next to all of these are Diogenids. So your land hermit crabs fit right in the middle of the tree of all the aquatic Diogenids. Right? It's not even a sister group to the outside. So what this literally means, based on these genes, is that the family, the genus Cenobita and the family Cenobitidae is just a derived branch in the middle of this massive Diogenid left-hand hermit crab tree. This is a gene tree, and you can make trees from genes. You can use 1 gene, you can use 4 genes. What they do is they sequence an animal, and it comes out all the different DNA letters in a row, and you compare them all, and the computer says these 2 are similar and joins them. This is the next most similar and joins it on and creates your tree that way. You can also generate your trees using morphological data. So you can say this guy has short eye stalks, or this has long eye stalks, and this guy's got a left hand, left claw bigger, and a right claw, and you can pull all that data, and the computer generates a tree and says all of these characteristics are closest between these 2 species, and then we include the next one. You make these trees of relationship. As you slowly step out, you're getting further and further away, and differences in their morphology. Fossil, you can also generate trees on fossil data. Fossils, of course, are only morphological because you can't get genes from fossils if they're just rock. But the good thing about fossils is it can show you certain types of morphology that you can compare to something living. So you could get a claw, right? Oh, I think I have a claw. Can you go forward? Here we go. This is the carapace, right? Or behind the eyes. Eyes will be here. What we call the carapace, or the shield, you may call it the shield, of a fossil hermit crab. Okay? And here's 2 claws of a fossil hermit crab, and the shell's gone, but the fossil has fossilized the mud that was inside the shell that was there. So literally, this is a hermit crab that has been fossilized and died in the shell it was in, and you've got to fossilize those claws right there. So this is indisputable evidence of morphology. You can compare this shield pattern to all the living hermit crabs if you want to, and all the fossil ones you've got. The only problem is fossils are very limiting, and not all parts of a hermit crab fossilize. For example, the soft butt on a land hermit crab is never been fossilized, right? The claws might. We have a ton of claws and we have a ton of shields in the fossil record for hermit crabs, and that's all we have. So it's limiting. But the cool thing is if a scientist can come along and they can date that fossil, find out how old it is, then you can then place a morphological feature in time. So that's the beauty of using fossils. You can say, oh, this particular type of shield is unique to the fossils, and this fossil is 65 million years old. If you can look at all the living hermit crabs and none of them have that shield, you can say 65 million years ago, hermit crabs looked like this, and they don't look like that now. Or if you can find a shield that looks like this on a living hermit crab today, you can say, oh my, that type of shield has been around for 65 million years, because you're able to put a date on the fossil. So fossils are limiting in that there's not a lot of them, and they have very limited morphology they can show you compared to a living crab in your hand. But the cool thing is, if you combine fossil data with living morphological data, you can actually put geological dates on features that you find in your living crabs, which is powerful. The genes, on the other hand, powerful tools because you're able to do multiple genes. Now, every morphological feature, every shield, every claw, every hair on a hermit crab's leg has been made from multiple genes inside its genome. You don't have a gene for that leg, a gene for that eye stalk, and a gene for that hair. There's multiple genes contribute to it. So when you have a gene tree that has 4 genes on it, you can go back one hand. If you go back to the gene tree here, it's based on 4 genes. These hermit crabs could have 50,000 genes. It's very limited in that regard. But the cool thing is, it does show you which hermit crabs share those genes more closely than others do. And so it allows you to place them in a tree of relationship, like we were talking about. So the ones all the way down the bottom here, right? These 3 down here, by the way, are the right-handed hermit crabs. So the gene tree says, see these 3 hermit crabs down here with right hands? They're very different to all of the left-handed ones here. So obviously morphology and genes should match, because every piece of morphology was made by altering the genes. Right? But we never know which genes do what. We still don't know that about humans. And we've done our genome. We have 27,000 genes in our body. But I can't tell you which gene does a particular thing in your body. We know for some of them, but not for all of them. Same thing for hermit crabs. So, and the distances here, so these long branches tell you that there's lots of change there. And when they are very close to each other, like here, it means there's very little genetic change. So things that are most closely related to each other, of course, going to have very little genetic change. Things that are very far away from each other, look at the length of these branches. All of the right-handed hermit crabs are a long way from all of these left-handed hermit crabs. But when you're in this genus Clibanaris here, you can see some of them have very, very short branch lengths. That means they're very close to each other. So your length of your branch tells you about relationships. Long branches, things have a very long history. They've been separated for a long time. Short branches, they have been separated for a very short amount of time. And that goes for no matter what data you generate your tree from, morphology or genes, it's the same story. The computers are generating those branch lengths according to the data that you put in, which also means that every tree is only as good as the data you put in. And so you put trash in, you get trash out, as we like to say. Okay? And the more data put in, the better trees should be. Every tree is a hypothesis based on that data, and everybody can argue whether a tree is good or bad. And we do. Scientists do that a lot. Generally. Okay, next. Next one. Okay, let's talk about symmetry now. I'm done with trees. I'm going to show you some more trees later, especially ones that are showing the relationships between all the species of the land hermit crabs. And so I'm going to remind you about those lengths of branches and things that we talked about there. These are all hermit crabs on the left-hand side. Here's your typical, here's your Dardanus, again, one of your left-handed hermit crabs. And you can see here that you've got his Burgess latro here. It has that bigger left-hand claw than the right-hand claw. This is a symmetrical hermit crab here. Its claws are about the same. Here's a left-handed one. Here's a left-handed one. Here's a right-handed one. Here's a right-handed one. This guy's a slightly larger right-handed one. And some of them are, this one's a slightly larger right. But this is kind of the fundamental difference between But you can also see that they nearly always have their butts always curl to one side or the other. They're fundamentally asymmetrical as an organism, with the exception of something like this guy here with a symmetrical hermit crab, which is about as symmetrical as you can get and looks as symmetrical as a crayfish or a lobster is. If you get a great big knife and cut one down the middle, you have almost two identical parts to it. Your typical hermit crab's not going to look like that because the back part of it is completely twisted around. But why would they be asymmetrical? Is another question for the audience. Why are they asymmetrical?
At least the ones with shells, it follows the curvature of the shell inside.
Right. They're limited by the shape of their shells. Grab the next slide. Gastropod shells, which is their principal home. 90% of all gastropod shells alive today and in the fossil record all coiled to the right. They're what's called dextrally coiled shells. The other 10% either have no coil or coiled to the left. They're called sinistrally coiled. Sinisterly coiled, right? Sinistrally coiled, left hand. And so the bulk of the shells a hermit crab is going to meet are right-hand coiled. And so all of their butts are right-hand coiled. Now, they can squeeze it into a left-hand coiled shell if they have to, because their butt's flexible. But you'll also notice that on their bodies, especially in the back of their abdomen, right, they don't have symmetrical structures there, because the inner part here rubs against the middle of the shell called the columella. It's that thing that runs down the middle of the shell that the spiral is around. So that's going to be right here, and that's going to be pushed right up against it. So you can't have structures like egg-carrying pleopods and stuff like that there, because it's pushed against the columella. You can only have them on the outside. And so a female hermit crab only has pleopods here and only carries eggs on the one side. And that's the outside that goes into the space and doesn't push against it. So it's made their entire back of their body asymmetrical to fit into those gastropod shells. Like I said, you can give them a left-hand shell and they'll fit into it. It's not ideal. Could be some great experiments to do. Give them left, give them right, see what they prefer. Give them a really nice left-hand shell, see if they'll take it over a grotty right hand, or if they'll naturally go for a right-hand shell. They're built to go into right-hand shells because the bulk of the shells they meet in the world are dextrally coiled and have been for nearly the entire fossil record of gastropods, which have been around as long as all of the arthropods and crustaceans have, 500 million years. So that's why they're fundamentally asymmetrical in that regard. And then on top of that, you have this left, right, and some of them being symmetrical. And that's because if you release them from the gastropod shell, then things like Burgess become more symmetrical, coconut crabs become more symmetrical, wood and bamboo living hermit crabs can become symmetrical, coral living hermit crabs can become symmetrical. They're no longer restricted by those right-hand shells. Next slide. This leads us to the weirdest hermit crabs of all. King crabs. Lithodes, as they're called. Lithodes, lithos means rock or stone. That's why they get called the stone crabs. Like I said, they're not the Florida stone crabs, it's a Manipi, which you eat, but Lithodes means the stone crab family. So Lithodes is the commonest genus, and that means a stone crab because they supposedly look like rocks. But they don't really. And you've all heard of king crabs before. You probably have eaten them before, right? Right, can I have the next slide? These are the crabs of the deadliest catch, right? This is what they're catching. This is what they're out there throwing those enormous traps over the side, catching hundreds at a time, right? And the true story, one of my undergraduate students was just a scientific observer on the deadliest catch boats. He got to see all of the nasty tricks that they get up to when they film stuff and things like that. So he came back with amazing stories of being on these catch boats. He spent a whole season recording the catches there. He's going back next season as well. So, so if we can go to the next slide. So this is a stone crab. It looks essentially, this is what we call a true crab, right? A brachyurine crab, like a stone crab, like a Dungeness crab, like, let me see if I can cover all the coastlines. Like a stone crab in Florida, like a Maryland blue crab, like a Dungeness crab, right? Cancer crabs, as they're called. They're all true crabs. They all have shells that tend to be wider than long, more spherical or oval, and they have symmetrical claws, approximately, usually fairly close to it, and they have 10 legs. And you'll count this guy here. We've got slightly asymmetrical claws. Remember, Lithodids are right-hand hermit crabs. And then we've only got 6 legs here. So we only have 8 limbs present here. We're missing 2. We're actually not missing them. They're tucked up underneath the abdomen. A little tiny pair of them. Remember the last 2 pairs of your hermit crab legs that are inside the shell? They're not out walking around. They're gripping the shell, holding onto it there, and they're much smaller than the rest. These guys, the 5th leg, they simply put it underneath their abdomen and fold their abdomen underneath, right? And it's in there. And they use it for things like cleaning their eggs and doing stuff like that, but it doesn't do anything. But it superficially looks like a true crab. So when mostly when you're eating king crabs, you're eating hermit crabs. They are a right-handed hermit crab that has become symmetrical because it's been released from the constraint of a gastropod shell. Why did these guys not use gastropod shells?
Deep water again?
They're deep water again, exactly. And they're also huge. Once you get big, you can't find shells. They're cold water, right, where there's not many shells. Deep water, where you can't make shells, right, and they're large. And so not much is going to eat them. A shell's not going to help them anyway. When big things eat them, a shell's not going to help them. So they are shellless, symmetrical hermit crabs. Lithodiid crabs. Well, if you eat snow crab legs, then you're eating true crabs. So just switch to snow crab. Okay? And don't tell crab industry that I told you. Okay. Next slide. Now, some of you, actually, I wanted to go. No, that's right. But if you look closely at a Lithodiid crab and you turn it over and you have a look at it, one, you can lift up the abdomen and see its tiny little legs. 2, if you look closely at the abdomen, it's not actually symmetrical. It twists slightly to the right. Okay? Just a little bit. It's kind of hard to see in this one. So in some of these, it's twisted slightly to one side. You can just see it on this one. If you drew an exact line down the middle, it just curves a little bit to one side at the end. So this is that asymmetrical, soft hermit crab butt that when you take it out of a shell, you've got to do something with it. So they've swung it underneath and they carry it here, and then they've calcified it again. And each of those sections that was soft and membranous has become harder, but it's still got this little tiny twist to it. It's not symmetrical like if you turn a Maryland blue crab over or a Dungeness crab over and look at the bottom side of it, that abdomen is completely symmetrical on the inside. So, and of course, they do have a slightly larger right-hand claw than the left. So they are fundamentally asymmetrical, slightly. But they're symmetrical. And if you look at where they fit in, the Lithodids and Haplogastrids, which are their close relatives, they come out of that big family group, the Paguridae, the right-handed hermit crabs. And in fact, they are derived from the Pagurids, and that led to, back in the 90s when they first did molecular work in this group, the hypothesis from a hermit to a king was the hypothesis, because they came from a hermit crab and became a symmetrical king crab, essentially. And that was disputed for a while in morphological evidence, but everybody agrees now that this is where they come from. They are a derived right-handed hermit crab that has become symmetrical and has increased in size and has become tasty. This means all hermit crabs are tasty, only the big ones are what you want to mess with.
Well, yeah, people say coconut crab.
Coconut crab is extremely delicious. It tastes like coconut. I don't eat hermit crab, as you all know. Okay, next slide. Now, some of you who were paying attention might have noticed when I showed you this slide here of all the different types of hermit crabs, there's one in the middle here that looks like a shell. And if you turn it over, it's got a little crab living underneath it, but it's not a crab underneath the shell. Its carapace has become a shell, like a turtle. Here, it's called Cryptolithodes, which means the hidden stone, the hidden stone crab. This is a king crab, a Lithoid crab. Its carapace has spread over the top of it, so it now is basically running around underneath the shell that it made itself. And it's got a little asymmetrical abdomen there, 8 legs, 4 pairs, and slightly asymmetrical claws with the right hand one bigger than the left hand one. So it's a special type of lithotic crab or stone crab. Okay. All right, so all of those are hermit crabs, even if they're symmetrical, right? Even if they're symmetrical deep sea pilochelids, even if they're symmetrical deep sea lithotids, whether they live in corals, whether they live in bamboo, whether they're left hand, right hand, whatever, they are all in the hermit crab group. And that, above the family level, we come to a level called the superfamily, which just means super above the family level, and they all belong to the superfamily Paguroidea. So your Cenobita genus is in the family Cenobitidae, which is in the superfamily Paguroidea. Okay? And they're all hermit crabs in there. And they're all in a group called the Anomura. The Anomura, ano means irregular, mura means tail, because everybody in this group has an unusual abdomen somehow. It doesn't fit the usual thing if you're looking at, like I said, a typical crab with a symmetrical abdomen. All of these guys have something that's different. So they were named because of that. And they were actually all thrown into a huge bucket because they weren't true crabs. And they've all remained in that bucket, but we've been looking at the relationships between them and to each other. This is where we get to the Adams family picture again. These guys look nothing like each other, but they are each other's closest relatives, which is really, really cool. So the Paguroidea are the true hermit crabs. They're in this group called the infraorder Anomura, and there are 2, 4, 6, possibly 7. There used to be a separate superfamily. Cenobita family used to be in the Cenobitoidea, but since been taken out and dropped back into here again. So it's back in the Paguroidea. So that no longer exists. So we have 2, 4, 6, 7 superfamilies in the Anomura. And I'm going to run you through each of these superfamilies, and you're going to have a look at what they look like, and you're going to see that none of them look like hermit crabs. Okay? First slide. Next one. So the first group we're going to go to is the superfamily Galateoidea, commonly called the squat lobsters, right? Because they have a shortened tail. Or if you're in the crustacean biology world, we call them squatties, right? Because they're called squat lobsters. And they all look like shortened lobsters. Symmetrical long claws out the front. Sometimes they tuck their tail underneath like this guy does. Sometimes it hangs out the back like this. Some of them, like this guy, look just like crabs. Okay? So we have the Galateas. This is the Galateas. There's a genus called Galatea, could be. This is the Munididea. This is the Munidopsidae. You can see they're all kind of similar to each other. And then this group here, the Porcellanidae, the porcellanid crabs. This is these guys' closest relative. He looks like a hermit crab. No, hermit crab. Looks like a true crab. Count the legs. 1, 2. Oh, he's missing a leg there. Sorry, left hand side. 1, 2, 3, 4. He doesn't have the fixed set of legs. Where is it? Tucked underneath his abdomen. He carries it underneath so you can't see it anymore. Just like a stone crab. He's flattened. Looks like this. Slips under rocks on coral reefs and stuff like that. And it is for all intents and purposes, looks like a little true crab. As always, count the legs. He's not. He is a crab-like Anomura, and he is most closely related to these weird squat lobsters. Okay, so this is the superfamily Galateoidea. Let's do the next group. Looking very similar is the superfamily Chirostyloidea, of which we have the Chirostylidae right here. The Eumenidae here, tropical, beautiful tropical squat. We also call these guys squatties. These guys are very colorful, very tiny, and many of them live symbiotically on corals and anemones and sea fans and sea urchins and stuff like that. They're beautiful little, they're often cryptically colored to look exactly like their hosts. And then this one here is a fairly new one, Kawaiidae. This is Kiwa hirsutum, right? The hairy Kiwa, also called the Yeti crab. This crab has no eye stalks. It's completely blind. They discovered it on those hydrothermal vents at the bottom of the ocean in the dark, right? So it's on those hot boiling water vents that have soluble minerals in the super hot water that comes out. And this guy, all of these hairs on his body, he grows bacteria on them, and he farms bacteria. He goes up to one of these vents and he sticks his arms over the hot water like that, and it flows up there, and all the bacteria, the chemosynthetic bacteria, they eat that liquid mineral that's coming out of there, and he grows millions of cells on all of his hairs, and then he just eats them off and puts them back in again. So he's farming symbiotic chemosynthetic bacteria on his hairy old arms. He has no eyes, no light down there, and he is a deep sea squat lobster, a deep sea squatty with no eyes. Kilo, as you know. Was that? Okay. All right. Okay, next one. All right, here's another group, the superfamily, the Hippoidea, right? Which includes one of my favorite families, the Hippidae, right? Which is the genus is Hippa, so it's Hippa in the Hippidae, in the Hippoidea. You can say all that quickly. So this is the Hippidae. Then you got the Albanais here, and then you've got the Blepharopodus here. These all burrow in the sand. You'll recognize this guy. This is your what sometimes called sand flea, sometimes called sand crab. It burrows on the coast, sandy surf coast, and they're found on both west and east and west coast of the U.S. All the parts of the tropical parts of the world you'll find them. And they're designed to burrow backwards into sand, which is why they're shaped like this. They have legs at the back that they burrow with, and the ones at the front that they use to, and they literally liquefy the sand and disappear down it. They can burrow 6 inches in about 2 seconds in wet sand. They're very good at that. And they're filter feeders again. These long antennae, just like that coral hermit crab, they stick their little antennae out of the sand while the wave washes over the top, and they catch all of that plankton, and then they duck down in the sand when the wave goes over, and they lick it all up again. And the next wave that comes in, they stick it up there, and then as it starts to go out, they pull it down and eat it, and the wave disappears again. And so sometimes you're standing there and you see this little antenna come up, and it pops down again, come up again, and down again, and every time the waves coming backwards and forwards, and all the shell seabirds are eating these guys because they're in the millions in the sand, in that wash zone on a typical sand beach. And as you'll notice, none of them look like hermit crabs. Okay? Next. And then we've got the last two groups here, which is really unique. The superfamily Agloidia, family Agloides. This is a endemic South American freshwater anamurian. It's the only totally freshwater anamurian. It's found in 5 countries in South America: Argentina, Uruguay, Bolivia, Brazil. Where am I missing there? No, that's true. Paraguay, thank you. Paraguay. Only those 5 countries. Lives in freshwater streams, has speciated there. They're now up to 100 species they found. And Chile, I'm sorry, Chilean coast. So I must have added one that shouldn't be there. They're on the Chilean coast as well. Only freshwater. They're not related to anything except this guy, which is superfamily Lomosoidea, Lomus, which is an endemic marine cold water only found on the southern coast of Australia. Nowhere else in the world. These two, in every single phylogenetic tree ever created, no matter what your data, are each other's closest relatives. And these guys have a fossil record. These guys don't. The only fossil ones we found of these is ones in New Zealand, in marine sediments. This had a marine ancestor, possibly related to an ancestor of this guy. They now moved from New Zealand to South America, left the ocean, moved into freshwater systems, and now have left no ancestors alive in the marine environment today. These are the closest. This guy, for all intents and purposes, looks like a crab. Count the legs. Okay, he's got a stiff little set tucked underneath the back here. Same with this guy here, little tiny fish set at the back. So even though they're crab-like, they are hermit crabs. Well, they're not hermit crabs. They're relatives of hermit crabs. They're in this group called the Anamura, the irregular tails. They are the most closely related crustaceans to the hermit crabs, and hermit crabs are in this group with them, but none of them look like hermit crabs. Okay, next group, next slide. So, several times I've mentioned it looks like a true crab, right? Carcinization. People heard of carcinization? There was this meme going on. It started in 2020, social media. I think everybody still looks like a crab. A little joke about it here. But they got their data from the scientists who said, oh my God, in multiple lineages of decapods, there's groups that assume a crab-like form. So we can go back and we can look at the lithoides, there's hermit crabs that do it. The galateoids, the porcelainic crabs do it. The lomasoidea do it. Right? And if you look at a whole bunch of crab-like forms, many of them are not true crabs, brachyurans, they're actually anomurans as well. So they think independently at least 5 times other types of crustaceans have assumed a crab-like form, which is the idea behind carcinization. It comes from Carcinus, which was the original genus for all crustaceans. When Linnaeus first started naming things, anything that looked like a crustacean, he called Carcinus to start off with, and then later we changed all names. For example, what's one that got changed from? No, I can't think of anything like that. But anyway, doesn't make a difference. So that's where that term comes from. Carcinization means to assume a crab-like body form, even if you're not a real crab. Okay? So we met many instances of that. Okay, so let's do a quick summary here. Land hermit crabs are hermit crabs. They belong in the Aguroidea, closest to the Diogenes. Right? So they are all, the hermit crabs in the genus Cenobita, in the family Cenobita, in the superfamily Aguroidea with the Diogenes, closest to the Diogenes, and they are anomurans with their relatives, of which none of them are hermit crabs, but they all have this weird irregular tail, and therefore they're put in this group called the Anomura. But where do the Anomura fit in to the crustacean story? Next slide. They belong to a group called the Decapoda. Deca 10, poda feet, the 10-footers. Okay? So they're 10-legged crustaceans. So here is a typical hermit crab, one of your hermit crabs. Okay? Once again, a few limbs missing, but here we go. We've got 1, 2, 3 main walking legs, 4th and 5th legs here, 5 on each side, 10 legs. And then you've got the abdomen at the back here. And so all hermit crabs, all anomurans, have 10 legs, even if you can't see them superficially on the outside. You know they've got their 4th and 5th tucked away or their 5th leg tucked away somewhere. Next slide. There are many crustaceans that have more than 10 legs, and ones you're very familiar with. There are anthropods, which live in freshwater and terrestrial environments. Sometimes you find them in leaf litter. They sometimes are hopping around. When there's seaweed on the beach, you can move it and things hop out of it. Sometimes called beach hoppers. Right? Isopods here, you all know pill bugs, sow bugs, whatever other common names you use to call them, roly-polies, etc. They are isopod crustaceans with multiple pairs of legs. The wonderful stomatopods, the mantis shrimps. Okay, all of these things under here are actually swimmerets or theropods, but it actually has 7 sets of limbs all the way up to its mouth there. And then this one beats it all. This is the most-legged crustacean. This is the Remipedia. This is a cave-living, aquatic, centipede-like crustacean that swims with multiple legs on multiple body segments. Right? It has a set of fangs at its mouth. It's predatory. It catches other cave-living organisms and eats them. They're about, you don't have to fear, they're only about an inch long at once. But they catch anything out there. And they have venomous fangs. They're the only venomous crustaceans known. Okay? Remember, venomous means you have to be injected. Poisonous means you have to eat it. Right? So we know there are poisonous crustaceans because they eat things. Some crabs eat things that they sequester the poison, and if you eat the crab, you die. Right? So there's lots of poisonous things. But venomous means you have to be injected. So this is the only venomous crustacean that's known. So these are all more than 10 legs. There's many crustaceans more than 10 legs. Next slide. All the decapods that you know best have 10 legs. the Caridians, the shrimps, in all their various forms that you eat. Okay? What are called the Astachidians, of which the crayfish fit into this group here. So usually they're freshwater, clawed, Astachidian crayfish. Then there's the Chelada, as they're now called, the clawed lobsters, of which the main lobster and the European lobster are the best proponents of it, including there's some smaller ones in Europe you may have eaten called nephrops, which are only half the size of them. Now we also have in this group, related to these, are the 8-chelate lobsters, and they are your tropical lobsters that you eat in the Caribbean that don't have claws. They have those 2 ginormous antennae with spikes on it that they carry over their head. They're extremely delicious, right? They belong in a group called the achilata, they're the non-clawed lobsters, and then you have the clawed lobster here, right? And then this is the only anamurans. We don't eat many anamurans. I could have put the Lithodes here, right? Lithodes, okay. But this is a bunch of squatties that they actually catch in big enough numbers in South America for it to be a commercial fishery. They swarm in the open ocean and in swarms of thousands, and they catch them in nets. And they're all, 6 inches max from tail to tip of the claw. And there's several species they eat down there. They catch them off Argentina, they catch them off Colombia and Ecuador, places on both coasts there. But there are some edible squatties. And then of course this is a true crab here that we eat many different types. This is a Southeast Asian mud crab that got mentioned today by the vet. Probably one of the tastiest crabs I've ever eaten, and some of the largest. A large one of these can feed 4 adults. Yeah, we're talking a planet. So they're really, really big and very, very tasty. So these are all decapods, because if you count them, they all have 10 legs. And many of them you eat those legs. You take the meat out of them, some of them you don't. Okay? So on to the next slide. So they all have a clawed limb being the first one, and then 4 sets of walking limbs that come after that. And then the abdomen that can be bent or not, that has what we call swimmerettes or pleopods underneath. The earliest fossil decapod that we have is about 360 million years old. It's called Paleopalemon, which means paleo, ancient, palemon shrimp, the ancient shrimp. Nuberii, this is what the drawing of it is like. This is what the actual fossil looks like. And you can see it's built like a shrimp or built like a lobster or built like a crayfish. All decapods, even including true crabs and hermit crabs, evolved from an ancestor that looked like this. 360 million. So put 360 million years into context here. All complex life evolved in the ocean about 500 million years ago. All the major phyla were represented, including the crustacea, 500 million years ago. And so 140 million years later, you've got the first decapods. The first dinosaurs appeared about 230 to 200 million years ago, so a little bit after that. And of course, the dinosaurs were all gone by 65 million years ago. So this is one of the oldest decapods known. And all those decapods I showed you would be derived from an ancestor that looked like this. Next slide. So all decapods, all the forms I showed you, everything I've showed you so far, was all present in the fossil record by 200 million years. So hermit crabs are at least 200 million years old, although the oldest one that we know of is a little younger than that. But all of these forms I showed you, all the anamurans, all the true crabs were present 200 million years ago. If you go back and look at this group called the Anamura, you find out that their sister group, the group that they're most closely related to, is the true crabs, those brachyurans. It makes kind of sense. They're all decapods. They're just slightly changed their body form. And they belong, this group is called the brachyura, which means the short tail. So you've got the irregular tail and the short tails. They're each other's closest relatives down here at the base of the tree. This is all of the true crabs in this branch. This is all of the Anamura I showed you, including the hermit crabs here, down on this branch here. And their common point of origin, sometimes this group is called the Mayura, which is, once again, it just means short tail. We keep repeating the same thing over and over again. They're each other's closest relatives, about this one here. But all of these forms were present 200 million years ago. Next. And we put another tree here. So here's the Mayura here again. Sister group at about 200 million years. All decapods were derived by about 360 million years ago. And we have the lobster-like forms here, and then we've got the hermit crabs and the true crabs here. These are some diagrams representing the fossils that we found. Eocarton is here and Platycota here. These are some diagrams of what they think the ancestral decapod might have looked like. And possibly fits right here at this 200 million year mark here. This is a good place to remind you that even though we have living crabs and hermit crabs, and I talk about them being each other's closest relatives, we have to remember that you can't have evolved from something that's living contemporaneously with you. So you can't say, oh, hermit crabs evolved from Maryland blue crabs. If they're both living at the same time, they have to evolve from something in the past. They can't have evolved from each other. So we can say they're each other's closest relatives, and their common ancestor was 200 million years ago and must have shared shared characteristics of both of the groups, because that's how evolution works. We think it may have looked like something like Eocaucinus here, but we can't be for sure, know for sure because you can't go back to them. You can find fossils and say, this is the most likely ancestor, but we'll never know. So just keep in mind, when we talk about evolution, you can't say something that's alive today evolved from something that's alive today. Right? There have to have been an extinct or ancient ancestor. Now, some of those ancestors may have forms that are around today that haven't changed. Sharks haven't changed their morphology in 200 million years. Crocodiles haven't changed. Crocodiles were around before the dinosaurs. They existed after the dinosaurs. They used to eat dinosaurs, and they're unchanged in all of that time. So morphology cannot change, but you don't say that dinosaurs evolved from a living crocodile. Right? So just keep that in mind. About living forms can be each other's closest relatives, but their common ancestors were in the past and must have looked something like them, but possibly not exactly like them. Okay, next. Okay, so let's do a quick summary here. Land hermit crabs are arthropods because they have hard exoskeleton with jointed limbs, right? Like a suit of armor. They're crustaceans because that exoskeleton and limbs is calcified and is mineralized. So, for example, the difference between a scorpion, a land scorpion, and a crab is that a land scorpion has a hard shell that's only made of proteins and phenols and stuff like that, while a hermit crab has a shell that's made of proteins, but then is mineralized with calcium carbonate and other minerals, and it becomes a hard, crunchy shells, but it becomes an inedible shell. You can pop a scorpion in your mouth, chew it up, swallow it, no trouble at all. Try doing that with a crab. You get a mouthful of minerals, which is what their protection is all about. So all crustaceans have a mineralized shell. It can be weakly mineralized, or it can be very strongly mineralized, but it is mineralized, and that's what separates them from the other arthropods that don't mineralize their shell. There's also some other subtle stuff going on there. Just to let you know, all crustaceans have two sets of antennae, right? The inner pair and the outer pair. Look at an insect, for example, which is not arthropod. You only ever have one set of antennae. Okay, look at that one. Then there's decapods, 10 legs or walking limbs, as we call them. Then the anamurans belong in the decapods. They have a modified abdomen, and usually limbs 4 and or 5 have been reduced, and they're not missing. They're often really small and tucked away somewhere where you can't see them. So most of the anamura look like that. Then within the anamura we have the paguroidians, the true hermit crabs. They all have that soft, twisted abdomen, with the exception of the Lethodus, which is still twisted, but it's less soft. Okay. And then within the paguroidians, they belong to the Cenobididae, which are the left-handed hermit crabs like the Diogenes, but they're the only terrestrial left-handed hermit crabs. Right? So that includes Burgess and Cenobita, and then Cenobita itself. These are the left-handed terrestrial hermit crabs that are totally dependent on a gastropod shell as an adult, which then separates them from Burgess. Not much else does. I mean, they do look different, but they are each other's closest relatives, and it's just the difference is size and the shell, really.
And sort of attached shell as bait.
Correct.
Right?
Absolutely. Yep, they all depend on shells as juveniles, but Burgess loses as an adult. So really, the only characteristic that separates Cenobita from Burgess is the lack of dependency on a shell of Burgess as an adult. All right. So land hermit crabs are unique, but only in their adaptations to terrestriality compared to the other hermit crabs. All the marine hermit crabs are adults. Next. So now for the last few slides, we're going to dive inside the family and go and look at Cenobita and look at some of the relationships in Cenobita. I pulled up a few papers, and I want to thank Stacy for sending me one recently, which I didn't have. Oh, no, I didn't remember the sheet paper, but sometimes you guys send me stuff because I'm just not looking these days, which is great. And I got a couple more recent papers, and we're going to see some new species that they're suggesting and have a discussion about relationships. So next slide. So back to the conventional family, which is the Cenobita. Right? These are all some of the different species. These are all most of the new ones that have just been published by She in 2021. But they're all in the same family. We've got multiple species. How many species do we currently have in Cenobita?
20 or
20
21.
to 21. Just a year ago, 16 to 17, right? So we just had an addition of 4. So it's actually a small number of species, a small genus compared to some of the other species of hermit crabs. And they're all terrestrial. Okay, next slide. So this is the tree that she and group put together in 2023. Once again, it's a gene tree. It's an odd shape that you can just change the way the tree's depicted. So all the trees I've been showing you are either vertical or horizontal. This has just been turned into a circular tree, all the same principle. And in effect, what happens here is that you don't have a root or an outgroup. Remember how we compared them to the right-handed crabs before? This doesn't have an outgroup like that. This is all Cedavino. We have different species here. We've got Brevimanus, Pseudorugosus, Violaceus, Spinosus, Cavapes, Pileatus, Purpureus, Lila, Rugosus, and Longivitus. Right? So these are all either Southeast Asian or the Indo-West Pacific species. For example, there are none of the American species here. No Curiatus, no Complicus. West African, something called Rubescens. It's not there either. Okay. Also, it doesn't have the endemic Australian variabilis either in there. Okay. So except for the C. brevimanus here, which is an East African one, right? This one group here. All the rest of these either have a large distribution that goes all the way from East Africa right across the Pacific, or they're specific to Southeast Asia. And you can see here that this brevimanus, which is an East African species here, separates from the rest of them here. And then you've got Pseudorugosus comes off here, and these are all individuals of the same species. Each of these numbers here represents one animal that they took tissue from. So this is all one thing. And then you can see there's two fundamental trees here, branches I should say. One branch here and another branch here with 4 species here and 4 species in that. So they're separated into 2 fundamental groups here. And this is only 10 of the 17 species that were known in 2023. So you know your crabs better than I do as living animals here. Is there any geographic information in this? Violaceus, Spinosus, this is the occurs in Australia too. Cavapes and Pileatus, that's another one that occurs in tropical reefs of Australia here. And I know that Lila and Purpureus, I think, are restricted to Southeast Asia. So which ones are these? Broadly East Africa to the Pacific, and which ones are just Southeast Asia? Does this represent geographic isolation in any way, shape, or form? I had a look at distributions of these, and it didn't seem like it to me. They're sort of like some of these that are perhaps limited to Southeast Asia, but some of these also have incredibly broad distributions from Kenya to Pitcairn Island, which is incredible when you think of the globe all the way across there. What we don't have are the northern Japanese ones. Is that right? We don't have
The furthest from
that's them here.
that's the Japanese.
Okay, so we do have a little plate with just the Japanese ones on there. What we don't have is the Australian and German variability. So interesting. Looking at genes, I also want to remind you, the shorter these little branches are, means there's virtually no genetic difference between this. And this is what you would expect. They're all individuals in the same species, right? So you should see short branches within a species, and then there should be longer branches between them if the genes are telling the right story. And the longer the branches are, the further away they are from each other. So this clade here and this clade here are not heavily separated from each other because these branch lengths are incredibly short. There is enough genetic information to separate them, but not as much as you've got between Brevimanus and Pseudorugosus here, which have very long branches. So these two here are definitely very genetically distinct from these 2 branches here. While some of these species within these branches, these 4 here, have pretty good separation. And the only thing is at the base here, you have very short branch lengths from each other. So you just have to remember that, and remember that we're talking about just a few genes, right, that are involved in this story. Can we see the next slide?
So those are individuals, right?
They are individual species. Each black number is an individual animal.
Okay.
All right, and this one's going to be much harder to see. This is the She's 2025 paper they brought out. They added 4 new species recently from Indonesia and the Philippines. Very much Southeast Asian endemic species that are here. Those 4 are added in the 4 different colors here. and now I don't have my notes, I can't even see what they say. That looks like granularis, I think there. Mollikensis, that's Sylvanus, and then Patsy. Okay, Patsy. So those four are the new species, and then variability. They managed to get hold of some tissue from the Australian endemic. All the rest of the black ones are exactly the same animals or sequences we saw in the last. Okay, so they took their 2023 paper, took all of the sequences from the 10, and then added these 5 new species to it. And then they just changed the tree. They put a root at the bottom here, out to, I think it's Brevimanus down here, and so it changes it from a circular tree to a type. That's just the case. But the interesting thing here is added 4 more species, so potentially 21 new species now in 2025. I want you also note that within some of these groups, like these 3 species here, the genetic differences are incredibly small. So they've recognized that there's morphological differences there. Somebody looked at them and initially thought, these are slightly different from each other. Let's sequence them. They got the genes from them, and they are genetically incredibly close. The distances are so small. People might argue if you didn't have the morphological evidence, some people would argue that they're all the same species. And this is where you can do that. You can be a lumper and say, no, all of these 4 belong in the same gene species, or you can be a splitter and you can say, no, they all have slight differences. We're going to call them something different. And you're entitled to your opinion, and then over time people will agree or disagree with you, essentially. But another thing I wanted to point out here is we have a couple of things are going on here. Geographically, we have variability here, and then where is I can't even read that anymore now. Is there. There's variability. Where's Spinosis now? Down here. There's Spinosis. There's Pilatus, there's variability. The 3 species that occur in Australia, and they're on 3 separate branches of this tree. Yeah. If you look at the tree structure, they're not each other's closest relatives. So now that begs the question about geography and relatedness. Normally, things that are closely related to each other should be close to each other, right? Geographically, because they're going to be speciating from the same stock and stuff like that. So my question to you is, why is this not the case for these species?
Continental drift?
That's very old. The oldest fossil, Cenobita, is 2 million years old from the Quaternary. Less than 3 million years old. Now, admittedly, they do not live in an environment that fossilizes really easily. Marine environments do, terrestrial environments do not. So it might not represent the oldest fossil, but they are a very young species. All of that continental drift stuff is going on in the last 250 million years and not necessarily in the last couple of million years. But we will talk about that in a moment, because we're going to talk about that when we get to the next slide. What about the life cycle of a land hermit crab that releases it from geographic constraints? They throw their babies in the sea. Say, good luck. Don't come back for your college fund. They throw them all into the ocean, and then they end up wherever the ocean currents send them. So no matter where the parents are, the babies are thrown out into the world ocean. So the geographic proximity may have less to do with speciation than changing ocean currents, or obviously currents will have to bring them back to where they came from, or the species will go extinct. There has to be some periodicity and cyclicity to currents and things like that. Otherwise, the parent population would die out and the babies would never make it back to that approximate area. But it also means that you only need a few babies to get sent somewhere else to start a new group somewhere else on a different, and you can get enough of them there, and the way evolution works, and enough time, you could end up with genetic and morphological differences. But here, we're restricted when we talk about terrestrial organisms normally that are reproducing on land. Usually geographic proximity leads to phylogenetic proximity. Your closest relatives are closest to you, in a way. This doesn't work this way because they're dumping their genes into the ocean, hoping they're going to end up somewhere. So that's why we don't see this geographic matching going on. Although there will be limits to that. Next slide. And that's where this slide comes in again. So here's that slide with all the different species on it. We've got species that go from here to here in the Pacific, from the Indian to the Pacific Ocean, pretty much all continuous through here. Some species go from here to here. Some are restricted just to here. Some are restricted just to here. So there's some endemism going on when they're restricted to a small area. And you can well imagine that you could mix all this group up here. The greatest diversity is probably here in what's called the coral triangle, where there's the greatest coral diversity in the world, the greatest diversity of coral reef fishes in the world, right, are in this coral triangle. And so in that tropical Southeast Asian triangle, this is probably the epicenter of Cynobita evolution. And they're probably coming out from this point and ending up in various places. And if they end up somewhere really wild out here, it's possible that they could become an endemic population because there might not be the currents to get their genes back into the pool, get their babies back into the pool here. And because they're restricted between 30 degrees south and 30 degrees north, I have a couple of questions like, how did this species get there?
I think it's from Spinoza. They look very similar.
Right. So Spinoza's
Is that from Kirk here?
is out here. But how does it get to Africa? It's got to go from this coast over here, right? Did it walk across Africa?
It's right on something.
Did it go to the, and if it goes to the Mediterranean around here, it leaves its tropical waters. It shouldn't survive. The larvae can't survive in waters that are colder, 30 degrees south and 30 degrees north.
And they get swim against the current.
And they can't swim against currents, right?
What about human agency?
It's possible. I don't know much. I didn't even know there was rubescence until I started researching.
It's not always how the world looks.
Well, that's true, but as I was saying, it was a long time ago that a lot of these continents were close to each other, and that actually predates any fossil evidence we have for Cynobita being present. Now, as I said, they live in environments that don't fossilize easily, like an open sandy beach, unlikely to leave a fossil on that. You live in a deep sea mud environment, you're more likely to leave your body there and it'll survive. So it doesn't necessarily say you will or will not fossilize. So a 2.5 million year old Cynobita fossil might be a very young one, and there could be some that are 100 million old. We just haven't found them yet. But they have a very young fossil record. And so we think they're a young group. We think. Now, Africa and South America used to be each other's closest cousins and actually used to be a continuous landmass. And that was 20 to 30 maximum million years ago. They started to separate, and this Atlantic Ocean got wider and wider and wider and wider. So it's perfectly sensible to think that this species might be related to the Caribbean species across an ever-increasing Atlantic Ocean. Right. So 20 million years ago, I can expect that. But so can it go both ways though? Predominantly, the currents come from Africa. Think of every hurricane that comes from Africa comes across the Atlantic to the west and then goes up north. All the currents do that below the equator, and then above the equator they go the other way over to Europe. And so going both ways across the Atlantic could be tricky. But then again, when it was closer, maybe the currents were different. We don't know. 20 million years ago, what was going on? But this is an interesting setup here, and I'd like to see genes of rubescence put into the pool. And in fact, there's no Clypeatus or compressus genes in any analyses right now. No one's done that. They're not all of those papers I showed you. It's all the Southeast Asian and Indo-Pacific. I would really like to see whether they're a separate group and if there is other closest relatives rubescent.
I do know something that has them.
Okay.
That's why I have pictures of it.
Okay.
We might over it.
All right. I know a guy who's interested in. And then here's the next story. We have a bunch of Caribbean species here, but there's also Eastern Pacific species spread from the eastern part of Central America down to Ecuador and possibly northern Chile here. Now remember, there's a Isthmus of Panama, there's land that's blocking it now, but 3 million years ago that was open ocean, and there was exchange between the two here. So 3 million years ago, it could be possible for this population to be continuous with an Eastern Pacific population. And so the species that are here, which I think is mostly Compressus, that's the East Pacific, right? And Clypeatus should be each other's closest relative with only a 3 million year separation history based on geological processes going on there. Now, then I would predict that if they're related to rubescence, then rubescence should be the next closest after that. then we get to this point here. But what about them? They made it all the way to Pitcairn Island, right? Like the Bounty, the HMS Bounty took all these people there, right? Easter Island's right about here somewhere. There's no record of them here. And then you've got this short jump over to the Eastern Pacific or the western coast of South America. But the problem is there, running north on that is the cold Humboldt Current, right? And all the waters around here are actually cold waters. You leave the warm tropical Pacific waters here, and this is all cold waters because you've got penguins swimming in the Humboldt Current, and giant squid, and all these cold water species are there. And it's not until you get up here above 30 degrees south where you meet the deserts and the warm waters of Ecuador and into Central America here. So it would be interesting to see if this ever happened. I would predict not, unless, of course, you had different arrangements of currents back in time. But it was going to be really interesting when you have all 21 species sequenced and morphological data to see what sort of history we have for all the species that are coming in there. And I would also predict that with closer looking here in the epicenter of it, we will continue to find more species, cryptic species. They're genetically this close and maybe morphologically this close. I mean, they're probably so recently speciated that they might have trouble separating each other, right? Because they're all speciated, moving on to new islands in that area there. So I would predict we get more species coming out of Southeast Asia here, and we would really like to know these relationships of the Atlantic group and whether it was possible, if there's any relationship between the species that are here and the ones that are on the South American coast. And I think that's it. There we go. That is it. So, once again, and I also wanted to, very quickly while I have the floor, I wanted to thank the people who have been supplying me with your treasured deceased individuals. Mary, Samantha, Dan, Holly Sedette, Shirley Miller, Allison Grant, Alicia Perry, Amanda Mitchell. Thank you. I have your specimens on my lab bench, and I'm going to be working with them, and you'll be credited with any work that comes out of it. So thank you. And I'm happy to take questions. And I can go back to any slides if you want.
What's happening with the lilas, with the larval lilas? Do you have the paper in?
It's very possible. My wonderful assistant, Anna Clark, unfortunately graduated. So now she's left my institution. Luckily, she's now working as an SEM tech at the Smithsonian Institution, which is cool. But I've now lost my SEM tech. She was doing all of that work, and she prepped them all, took pictures of them all, gave me the file with the hundreds of pictures, and I haven't had a minute to look at them yet. But I have got Lila images of all the stages you sent for the sea Lila.
That recent paper that Stacey shared and I shared with you was much more condensed. I noticed, if you want me to go back over my notes, like they were like, it ate pork, that's the megalopa, and I was like, well, they had, this type of pork, and this, like maybe I gave you too much information. If you want me to,
I'll need to digest it first. I need to find the time to sit down and look at it. I know that before Anna left and graduated, she gave me everything she did, and knowing her, it'll be super organized and folders or whatever she gave me. And I'll look at it, and I have all the original notes she sent me, and that'll be on my background to get to, because that's too good a set of data to not do something which sounds fine. The question, Walter?
Yeah, my question is, so most of the land hermit crabs are left-handed, correct? Right-handed.
All of them, I think.
Okay. So there are right-handed marine species, and as you said, 90% of the shells that you find are of the right-handed, like the shells.
Yep.
Then you get left-handed, like I know, for instance, the lightning bug is a left-handed opening shell that's found in the Florida Keys.
Right.
And a lot of the times, I've stopped watching because I can't watch them anymore, but I used to watch shell collection videos, and a lot of the times they would pick up the lightning whelps and they would be inhabited with marine hermit crabs. Do you suspect that the few that are in that sort of right-handed branch of marine hermit crab involved because like of scarcity of shells or like because they were like, hey, there's not enough for all of us to be in this certain type of shell. I'm going to involve so I can inhabit a shell that's bountiful but doesn't have so much competition because I go the other way.
I agree. We have to remember that the handiness doesn't have anything to do with the shell structure because they're not putting those claws inside the shell, not all the way.
No.
It's the back, it's their butts that are specifically designed for the 90% of all the mollusk shells that are right-handed. So they can be left or right-handed and still be in right-handed shells.
I just didn't know if the aperture was like stop them from.
They might be limited. We know examples of crabs that modify their aperture. Do your land hermit crabs do that? Do they rub the aperture edges to change the shape of the shell? Right. We've actually found fossil shells with these scratches on them that we know were made by hermit crabs. The hermit crabs are not in the shells anymore, but we know that marine hermit crabs modify the architecture of usually just the opening of the shell they're in to suit them better. And that could be whether you're a left or right-handed crab in a right-handed shell or not. But the actual asymmetry of the butt is all to do with the shell structure and would limit their ability to breed, but not their ability to live in the shell. And my guess would be if you're a female and you find a left-handed shell, you have got no room to put your eggs on your pleopods in the left-handed shell. So you're going to get out of that before you can breed. You have to find preferentially a right-hand shell. And basically they're all right twisted because the bulk of the shells they run into are like that. And they can live in the left-handed shell. Like, you might be uncomfortable, but their body's flexible. They can move the other way. But their reproductive system is not built to do that. And in fact, their internal reproductive system, the ovary is 3 times the size on the outside than it is on the inside. So they have twinned ovaries, but the ovary is little tiny and short on the inside and 3 times as long on the outside. Same thing with the male testes. The entire internal system is asymmetrical because of having to twist into those dextral shells. But the handedness is not necessarily associated with the shell type.
Do right-handed crabs have right twisted paths? Or can you go the other direction?
No, they all have, whether you're left or right-handed, you have a right twisted path if it is a dextral shell. So it's actually, there's no relationship between left and right-handed and the shell type that they're in, unless you have a large claw. Now, some crabs, their large claw, the end of it, when they pull it in like a door, and it forms an operculum, and it fits exactly into particular shell species types. One of the Pagura species on the eastern Atlantic shore here, I don't remember which one it is now, so Polcaris or Longomanus or the other. The claw is expanded, and when it pulls into a particular shell, it's the last thing you see, and they literally block their shell with their massive claw, and it has to be almost exactly the same size. So they preferentially find the shells that their claws will block off. If you have a shell with a long aperture, they're not going to get in because they can't effectively close it. So there might be some limitations on the shell types depending on their left and right-handedness. But there's not going to be, it's not going to be linked to the left and right-handed, because most of the time, all of that part of their body is out of the shell and not back inside. Make sense?
Mm-hmm. Archyliatus are almost like that.
Yeah.
I've noticed, and they also have what we call a shield leg that has no setae on it, so there's nothing to grab, right? It's just this slippery thing, but when they close, they get pretty tight.
Yeah, and remember, it might not necessarily be to predation too. It could be to keep somebody from evicting you from your shell. So it could be to protect your home, not just to protect yourself from predation. All those sorts of things. Yeah.
For the ones that lived in the coral and or with an anemone, do they have a mutualistic relationship? The crabs and those?
Those sorts of relationships are hard to define, most likely mutual. You imagine if you're a deep sea anemone, anemones usually need something fairly solid to attach to. And if you're living at 6,000 meters on a very soft, muddy floor. There's probably not much to attach to, so it's mutually beneficial to stick yourself to a hermit crab or be stuck to one. And then he transports you around to new food sources, fresh water. And though you notice those deep sea crabs have incredibly long legs because they spread themselves out, they have to walk on soft mud. And so they're incredibly long limbed and they lift themselves several inches off the mud and walk along it like spiders. And there's this little anemone who would normally have his head down there and all that mud getting stuck to it, lifted 6 inches off the bottom, being transported around to food sources. So I can well imagine that there's benefits to both, but you have to be careful how you define mutualism. It looks like mutualism to us. The anemone could be totally pissed that somebody took it off the Titanic and carried it around the rest of the ocean. You know, we just don't know. It looks like it's mutualism. Yeah, exactly.
And then what about with the corals? What about with the corals too? Or is that more?
I can't imagine that the coral can't grow where the crab is. It grows around the crab, and the crab actively stops it from expanding its body into its cavity, and probably pinches off the tentacles that try to include it there. So most likely not. But once again, does a hermit crab, is it a messy feeder? Is it dropping bits of plankton off its feelers onto the tentacles of the corals, and they're like, yeah, thanks very much. We really can't say what's going on there. Once again, it doesn't look like mutualism, but we're not sure what's happening. It's possibly so. They're both filter feeding. They're both after the same food sources. Correct. So it might even be antagonistic, not necessarily. Yeah, really hard for us to say.
You may have, I missed the beginning because I was troubleshooting, but did you talk at all about horseshoe crabs? I know they're not crabs, but they're very ancient. Like, how does that, where do they fit in?
Where do they fit in? They're arthropods. They are not crustaceans. So they are aquatic scorpions, essentially. Their nearest relative is probably something like a terrestrial scorpion. It probably was the ancient marine scorpions that were called eurypterids. That's probably their closest relative that's now extinct. And because they're extinct, the terrestrial scorpion kind of becomes their homologue on land. And that, by the way, the terrestrial water scorpion, terrestrial marine scorpions were the largest aquatic arthropods. Some of them were 7 to 8 feet long, bigger than man. And we have great fossils of them in many museums, but they were the largest aquatic arthropods.
And did they have that traditional shell?
They had the same body divisions as a horseshoe crab has, and very similar limb structure. They just didn't have the carapace extended out into that overarching shell.
They almost look like that one stone crab that you showed us, where it's like they created the shit. Like I was like, that looks like a horseshoe crab.
That's just convergent evolution. How can I protect myself in a marine environment and stop somebody from coming down and biting my eyes and so if you can grow your shell out over the top of you, you can protect all your legs and all your gills and all that sort of thing. So it happens multiple times in many different groups. Yeah, absolutely.
Does that mean that the horseshoe crab shell is not mineralized?
Correct. It is not mineralized. It has, it's got things other than proteins in there, but it is not a calcified shell. They do something called tanning with it. They do phenolic tanning to the proteins that are there, which makes it turn dark, black or brown, which is why most insects and spiders and scorpions are black or brown. They tan the proteins that are there, and phenolic tanning leads to hardening and makes it hard. Also makes it slightly brittle, but that's what's happening there. But they do have some minerals in it. They just don't mineralize it like crustaceans do, but predominantly calcium carbonate, which gives it that white chalky appearance.
Sergio had a the calcification, right? So you're saying that snails or whatever, the hermit crabs cannot calcify at deeper sea levels, right? They can't make their shells, therefore they don't live at that. So but the crabs can.
The crabs can. Crabs have more protein in their carapaces than a snail does. A snail shell is predominantly calcium carbonate with small amounts of protein in it. A hermit crab shell is probably carapace is predominantly protein with a calcium carbonate matrix on it. So I I bet they're less calcified at depth than a tropical shallow water hermit crab is, but they're still, and also less calcified than a typical snail shell is. And they can protect that mineral, they can protect the calcium carbonate inside their protein matrix, while a snail shell, which is just bare calcium carbonate, slowly erodes essentially at depth. Yeah.
Sergio had asked in chat, could you share your insight on Patagoras Rex?
Patagoras Rex. I don't think I know Patagoras Rex. The new hermit crab species? I don't know. No, Pagurus. Is it Pagurus Rex? Patagoras Rex? Oh. I actually haven't heard of Patagoras. It could be a new species of hermit crab I haven't heard of. It could be a Patagonian Pagurus. Pagurus maybe? Pagurus. I actually don't know that. If you could provide more context, I'd be happy to think about it. It may be something I haven't come across.
I think it's spreading out, right? We're discovering more and finding more.
And it's becoming cheaper. You can do an entire genome for $1,000 worth. It cost millions when we did the human genome back in the 90s. It's becoming incredibly useful. Genomics is revolutionizing biology. Yeah. I mean, the talk today from the vets is really interesting, and I think where you need to go for that, as I was talking to somebody today, is the genomic route. So you swab your hermit crab, you swab inside the shell, you get a list of all the organisms you find there, and then you have a library of what a healthy hermit crab has on it, and then when one comes in sick, you swab it. You do it, and you compare the two libraries and say, oh, look, the sick one's different in these areas. It's missing this, or it's got an addition of these. Then you can start to think about the microbes that might be causing problems. It's going to revolutionize.
You talk about taxonomy, just as a side note, not hermit crabs. I was watching about dinosaurs and fossil apes last night. We've always thought dinosaurs and the existence, they will die off. But now the more common theory is that feathered dinosaurs, birds, are still dinosaurs. We still do have living dinosaurs. Crocodiles.
We are so lucky to live in a world of feathered dinosaurs.
Right.
10,000 species, 11,000 species of feathered dinosaurs. And they come and they sit while you're having a beer and let you watch them.
Yeah. Or live in your house with you.
Or live in your house with you, right? Poop on your lawn. It's the most fabulous thing ever. Yeah.
And then you go back to Darwin, everything's visual. Like, this animal look the same, what characteristics.
Yeah.
Genetic data that shows, oh, no, they may look alike, but they're not, or they don't look anything alike, but they're very alike.
Yeah.
Actually, that's what I was thinking of when you were talking about the different species in the Indo-Pacific, where they're very, very closely related. Like you start thinking about Darwin's finches,
Yeah.
where they're similar, but separated by different islands, and each has developed their own specific genomic tag, and like how with having a species that starts an aquatic lifestyle that's very much based on where the current has taken them, that you can have like thousands of species that are very closely, like you would pick two up and say these are the same, but then if you genome them, they're different.
Yeah. And that we're terming that now cryptic species, and that's what everyone's looking into, where we used to think there was one species of blah that went all over the world, people saying, is that possible? They look exactly the same all over the world, and then we look at it genomically and we see the differences. So we've actually got cryptic genomic species in there, even though it might be almost impossible to tell them morphologically. Because remember, you can change genes and not change morphology very much, right? Especially if you just change a few genes. It takes a long time to change morphology from genomic changes, and they don't necessarily have to be one-to-one changes.
Even in humans, like you were saying that like we're powerful with, like, I work in pediatrics, and like we're doing a lot of microarray panels now for children that like come in with developmental delay or autism and stuff like that, and like they come back and they're like, we found that this kid's got a gene that's different. We don't know the significance of it. Like, they can't study every single gene, but like, then they're sent on, to speak with a geneticist, because, each little piece can alter us. It doesn't necessarily mean that we're outwardly different, but, and it's so interesting.
Yeah. I want to come back to talking about genomics and taxonomy, because in 1758, when Linnaeus came up with his system, they only knew a few thousand organisms back then. We now have millions. But interestingly enough, it was totally based on morphological differences and similarities. Linnaeus was a religious man, and he was doing it to document the biodiversity of the world to glorify God. He was doing it for religious reasons. And it's interesting that it only took 100 years later, in 1858, when Charles Darwin was publishing his Theories of Evolution, he was still using a Linnaean system that we're still using today, right? And 90% or more of Linnaeus' system still stands, because if two things look alike, they're more likely to be related to each other than not. There's only a few exceptions, like these convergences that are they're exceptions, and they're a tiny amount. And so two things that look almost the same are more likely to be related to each other than not. And so the system still stands up 200 years later, even with genomics. We're changing very little of life's tree because of that. It was fundamentally put in place 200 years ago.
So, yeah, genetically they're slightly varying, but not.
Yeah.
They're not drastic.
The mysterious things I mentioned before are the things that haven't changed morphologically for 300 million years. It's like, here's a great model, I change it. But of course, we don't have genomes to compare a shark from 300 million years ago with shark today. The genomes must have changed, but their morphology is virtually unchanged. Same with crocodilians.
The great white fin is a primary shark. It has not genetically morphed or anything changed. That's what it's been for millions of years. But why? It doesn't have a predator, doesn't need to change for predation other than man, obviously, right? So it hasn't needed to change because it's an alpha predator, right? You can't stop it. It can live and eat anything. Yeah. So it doesn't need to change. It's agile, right? So it's quote unquote perfect, right?
Yeah, well, so they're saying. But yeah, no, it's really interesting. You know, we have so many groups of which are identical. Horseshoe crab, 250 million years, it's looked exactly like that. Sure. Ferns, another great group, crocodilians, turtles. We had ancient sea turtles that looked just like leatherbacks, and they were millions of years old, and they haven't changed. So some groups, you know, stay morphologically, and they have to be genetically different, but they stay morphologically unchanged. And in other groups, in 2.5 million years, you can make 20 species out of one, you know, things like that. It's really interesting. It sort of shows you the adaptability of the of life and of the mechanisms of natural selection and evolution. They either run rapidly or they can slow down. It all depends on the needs of the organism. But I didn't want to go into an evolution.
That's fascinating how, rather than changing the lineages, we're just refining them with genetics.
To some extent, it's exactly what we do. We find a few things that, you know, like there's have you ever heard of a tongue worm? It's a parasite that gets into the mouth of vertebrates, like snakes and stuff like that.
I think it's a fish.
That's an isopod. It's actually a crustacean. It replaces the tongue. It eats the tongue and sits in the place where the fish's tongue would be and acts like the fish's tongue afterwards. But it's permanently attached to the fish's large brain. What does it do for it? It gets to eat the tissues and drink the blood of the fish. So it's like a vampire. And it just sits in its mouth. And the fish just uses it as a tongue for the rest of its life. So that's a nice one. But anyway, the tongue worm, it's found in snakes a lot. It's found in reptiles. It's found in a few mammals like foxes and stuff like that. And it's just this yucky looking flat worm-like thing that's in the mouth of terrestrial vertebrates. And for a long time, it was a group all of its own. And then we sequenced it, and it's a crustacean. It's a crustacean that must have moved out of an aquatic habitat into vertebrates when they moved onto land, and then has a life cycle there. Because when it hatches out of its egg, all crustaceans have exactly the same first stage. It's called a nauplius. All crustaceans have this thing, no matter what you are. Whether you're an isopod, an amphipod, a hermit crab, your first thing that hatches out of it is called a nauplius. And the first stage that hatches out of a tongue worm's egg looks like a crustacean nauplius. And then in the nose of terrestrial vertebrates. But that's where genomics helps. Because, well, it has no morphology like a lot of parasites. If you live inside somebody's gut, you don't have to look like anything. All intestinal parasites look like worms. Whether you're a crustacean or whether you're a nematode worm or a flatworm or whatever, you just look like a white worm because you don't need any morphology if you're going to live in the cold, dark intestine of anybody. And so you can't use morphology, so that's where genomics has revolutionized a lot of that. When there's morphology absent, then genomics has been incredibly good. Yeah, but that was a huge, when people found that, it was just astonishing. And then they went back and looked more closely, looked at eggs, looked at life stages, looked at things like reproductive morphology. For example, the sperm structure of a tongueworm looks like a crustacean sperm. It's just incredible. So, yeah, so you look a little more closely at morphology.
Okay, now I'm curious. How does a crustacean sperm vary from like a male sperm? Like,
So now, you're now entering the realm of my 40 years of research on crustacean reproduction, where I spent my entire life looking at sperm cells of crustaceans.
like we're all accustomed to like head wiggles.
Exactly. Get unaccustomed to that because all crabs are just head and there's no tail. And they don't swim. And so, but the fundamental parts of the head are there. You can see the mitochondria and you can see the nucleus and the genome there and everything. They just don't have time to swim. And then within all swimming sperm, every major group has a different morphology depending on what the reproductive biology is like. If you're just going to put your sperm in the ocean and let them swim around in the water, they have to be built very differently than if you put them inside a reproductive system and they have to swim through mucus because the engineering, the energetics, and the physical environment is different. You have to build a different sperm for different fertilization environments.
Yeah, because they don't have to. There's no acid and secretions.
Yeah. So if you're a sponge, you produce a sperm. It swims for 15 minutes in the ocean. It might travel a couple of meters through liquid water, which has a certain viscosity. You can make a very simple little swimming machine, but you can't put that sperm in the mucus of the female reproductive system and expect it to go anywhere.
No.
But so you have to design a sperm that can swim for 24 hours in mucus, and so you have to redesign the sperm structure. Yeah, sorry. I'm going home now. As Karen said, don't get me started on sperm, all right? Oh, okay.
Do we have a sperm count, like an average sperm count? Because we know that females can have like 10,000 eggs. How many sperm are in a typical sperm count?
There is some published information on that. My bet it would vary depending on the size of the male because the investment has to be a proportion of their body mass. Larger males can produce more sperm. More spermatophores can fertilize more females. That's why large body size gets promoted. They can be competitive. They can compete other males. They can grab any female they want. And the bigger their body mass, the bigger proportion of their energy they can spend on that. So if you're your first year male and it's your first year as a reproductive male and you're only this big, there's only a certain amount of energy you can devote to it, and you're probably not going to get access to a female anyway, right? And so you maximize your body size and making big claws, and then you can become competitive. But it would vary depending on body size, age.
But I mean, it's so
And same with
different
females.
in men and women, humans. It's like
Correct.
1 to
Correct.
gazillions. Like,
That's just a fundamental difference between what males and females want.
so do you think it's 1 to 1, like
No.
Hermit crabs?
No. It's always millions to hundreds or thousands. Tens of millions or hundreds of millions to thousands is how it works. And it's a fundamental battle between, females make a huge investment of energy. An egg is already thousands of times bigger than a single sperm cell. They have to carry them around on their body. They have to sometimes do parental care afterwards. They have to devote their energy, like milk and food, to their young. It's a huge energy investment. For the males, each individual sperm cell is 1/800 millionth of their energy investment, and they only need one. Right? And so for them, what's that? Yeah, every sperm is sacred, don't you? Mighty priceless. Yeah, and so males want to maximize spreading their genes around, and they can do so because it's a very small investment. They actually do a greater investment in their reproductive morphology, bigger claws, bigger horns, bigger muscles, all that sort of stuff, than they do in sperm production, because it's all about access to female, and the females need to be choosy. They're making a huge investment in comparison, and they need to be very choosy about what they do.
We see guarding up to 3 days.
Yeah. Yeah, and I mean, it's all different. Oh, here's another really interesting thing about reproduction in hermit crabs. All aquatic hermit crabs, right? There's no internal fertilization at all. All true crabs, a male puts the sperm inside the female system. In a hermit crab, their closest relative, all aquatic hermit crabs, the male puts the sperm in packets on the outside of the female. And then when you move to a terrestrial environment, that has not changed. They haven't had it long enough to change that system. And so they're still sticking sperm in a bag on the outside of a female. It's in air, it can desiccate, right? But they still have the same mechanism as they had as their aquatic relatives had. It obviously works. We have babies, but there must be some fundamental differences in, for example, how it sets. It may be in a gel or whatever, and we have like this much information about that. So help me with that.
For all we know, the female could straight off.
The female could be doing female choice. She's like, I wasn't interested in that male anyway. Exactly. And where there are multiple males that can have access to a female, they can be very indiscriminate about where they're putting their spermatophore packets. And I've seen photographs of females with sperm on their shell, on their head, on their floor, and she's staggering around after the one with the other down. But none of those are functional. Those males are wasted now, really. And so I think there's a female choice going on there. Less so than if it was internal fertilization, where the female literally has to allow the male to do that, and there has to be this mate guarding stuff going on. There has to be a lot of very complex chemical communication and behavioral communication going on, because the males can't just walk on by and here's a sperm packet, see you later, right? So there has to be lots of stuff, but it's really interesting that they have exactly the same reproductive biology as their diogeneid aquatic relatives. They haven't been separated long enough from them to evolve a different system to that, because it doesn't seem like the best system for terrestrial fertilization, but it works. And they are keeping a moist, wet environment. The female's keeping a moist, wet environment, so it is possible. But there has to be modification going on there at the cellular level to allow that to occur. And there's nothing known about it. Yeah. Totally cool.
Have we warmed you out?
I'm ready for a beer. Thank you. As I said at the beginning, it's really a great pleasure for me to come here and do this and see all my friends and to share some knowledge with you. And I learn something every time I come here. So it's a mutualism, okay?
Well, we appreciate you so much.
Break. Thank you so much.